Hello everyone, welcome you all in this video lecture. So in this video tutorial, we will discuss about the gram-negative bacteria, their cell wall composition and about the effects of gram-negative bacteria. So let's start with the introductive part. So before moving to the main topic and understand about the gram-negative bacteria, let's discuss some basic facts and characteristic features about the bacteria. So the bacteria are one of the oldest life forms in the earth, which means that these cellular organelles are considered as one of the oldest or most primitive cellular organisms found in the earth according to the evolution theory. It is considered that they are one of the most primitive organisms in the earth, and they are prokaryotic and unicellular organisms. So basically the bacteria are very small in size, which cannot be seen by naked eyes. So these organisms can be only seen with the help of electron microscope, and that's why they are also called as microscopic organism. Now the cell composition of bacteria is prokaryotic in nature, which means that the bacteria lacks several cytoplasmic organelles like mitochondria, endoplasmic reticulum and many other membrane-bound organelles which are considered as cytoplasmic organelles and found in the eukaryotic cells. And these prokaryotic cells also lack a well-defined nucleus. That's why they are considered as prokaryotic organisms. Now, the bacteria are categorized into several types on the basis of their shape, their cell wall composition and oxygen requirement. For example, according to the oxygen requirement of bacteria, they are divided into two types, which are aerobic bacteria and another one is anaerobic bacteria. Similarly, on the basis of shape, they are also divided into different types like cocci, bacillus, spiral and many other types. So according to the cell wall composition of bacteria, they are divided into two types, which are gram-positive bacteria and the another type is gram-negative bacteria. So in this video lecture, we will discuss about the gram-negative bacteria, their cell wall composition and their various effects in humans and other higher animals. So let's understand about the gram-negative bacteria and about some characteristic features about these bacteria. So basically they are the bacteria which define the opposite of gram-positive bacteria, which means that they are the group of aerobic bacteria mainly, which do not retain crystal violet color in gram staining. So the gram-negative bacteria will lose the crystal violet dye color after a wash with alcohol and take up only the pink or red color of the counter stain, which is safranin. And that's why they appear red and pink color under the microscope, and that's why they are considered as gram-negative bacteria. So these bacteria are normally found in the gastrointestinal tract of higher animals. Some examples of gram-negative bacteria includes Vibrio, Pseudomonas, Campylobacter and many other bacterial organisms. So these gram-negative bacteria are found in the wide range of environments. They have life ranging from normal flora to the pathogenic bacteria. So let's discuss about the gram staining method and understand some basic facts about this method. So basically this is the most crucial staining method used in the field of microbiology to differentiate between the different species of bacteria. And this method was first discovered by Hans Christian Gram in 1882, and the method was also named over it as gram staining method, and the bacteria are also called as gram positive and gram negative according to the result and according to the color which can be seen in the electron microscope. So this method involves the use of crystal violet and methylene blue for the staining and also use safranin for the counter staining. So in this method, at the first time the crystal violet or methylene blue staining done in the microscopic organisms or in bacteria, and after that it was washed by the help of alcohol, and then the counter staining was done by the safranin. So what happens that the organisms who retain the primary color and appear as purple brown under the electron microscope are called as gram-positive bacteria, which means that gram-positive bacteria retain the crystal violet color and it did not wash by the alcohol. And the organisms which do not take the primary staining, which is washed out by alcohol and appear red or pink under the electron microscope by the effect of safranin, and they are called as gram-negative bacteria. So this differentiation is due to the different cell wall composition of gram-positive and gram-negative bacteria. Now let's understand about some characteristic features of gram-negative bacteria. So basically, as we have discussed, that there is a cell wall differentiation between gram-negative and gram-positive bacteria. So the cell wall of gram-negative bacteria is made up of thin layer with an outer membrane. But in the case of gram-positive bacteria, they have a thick layer of peptidoglycan and they do not have any outer membrane. So the gram-negative bacteria contain a higher content of lipids in its structure and a very less amount of muramic acid, which are also called as peptidoglycan. And they also contain some lipopolysaccharides and sialic acid in its structure. Now the gram-negative bacteria are sensitive to streptomycin, which is a class of antibiotic. So these bacteria can retain or can survive with the effects of antibiotic streptomycin, or the antibiotic streptomycin cannot affect the cell wall or the structure of gram-negative bacteria. Now let's discuss about the cell wall composition of gram-negative bacteria. So the cell wall of these bacteria have a unique cell envelope or an outer membrane which is not present in gram-positive bacteria. Now the cell wall of the gram-negative bacteria is composed of 3 layers which have an outer membrane which is called as an envelope, and in between a thin peptidoglycan layer or a layer of muramic acid, and the third layer is of cytoplasmic membrane. And there is a space between the cytoplasmic membrane or the plasma membrane and the cell wall, which is called as periplasmic space. So now let's discuss about the different components of cell wall of the bacteria. So the first component is periplasmic space. So basically in the case of gram-negative bacteria, this periplasmic space is larger as compared to the positive bacteria. And this space is made up of different types of proteins, and these proteins are helpful in acquiring several nutrients for the cell and also helpful to acquiring hydrolytic enzymes also. So they also have some enzymes which can synthesize the peptidoglycan, which is also a component for the cell wall, and they also modify several toxic elements to prevent the bacterial cell. Now the another component of cell wall is peptidoglycan layer. So in the case of gram-negative bacteria, this peptidoglycan layer is very thin. It is found in the above the plasma membrane or the cell membrane, and sometimes it not found in the bacterial cell or in the gram-negative bacteria. So the gram-negative bacteria have a very thin peptidoglycan layer which make about only 5 to 10% of the cell weight. And sometimes like in E. coli and another some genus, they have only 2 nanometer thick peptidoglycan. The another component is the outer membrane, which is the specific feature of gram-negative bacteria. This outer membrane, which is also called as lipopolysaccharide, is composed of bronze lipoprotein. These bronze lipoprotein strongly links between the outer membrane and the peptidoglycan layer of the cell wall, and they also provide some adhesion sites to the outer membrane, which play a role in allowing cell contact and membrane fusion. So the outer membrane is majorly made up of lipopolysaccharides, which are commonly called as LPS, and they are large complex molecules which are made up of lipids and carbohydrates. So basically the lipopolysaccharide is made up of three components or three units, which are named as lipid A, the core polysaccharide, and the outside chain. Here the O side chain is also called as O antigen that extends from the core outwardly. Now the lipopolysaccharides are also responsible for protecting the cell wall from external attacks, and it is a porine structure, and there are pores present in the lipopolysaccharide which allow some nutrients to enter into the cell. So let's understand about the cell wall composition of gram-negative bacteria with the help of this diagrammatic structure. So here we can see this is the structure of gram-negative cell wall having different components. So in the outermost layer it is the O-specific side chain, which is also called as outer membrane, made up of O side chains, lipopolysaccharides, and also lipid A molecules. So there are the porins present in this membrane. This is the outer membrane having specific side chains, and they are the porins present in this membrane. So between the cell wall and the plasma membrane of the cell, there is a periplasmic space which is comparatively larger in the negative bacteria as compared to the gram-positive bacteria. And there are Brown's lipoprotein molecules which attach and make strength between the outer membrane and the peptidoglycan layer of the cell wall. So here we can see this is a very thin, only two-sheeted peptidoglycan layer present in the cell wall. So now let's understand about the effects of gram-negative bacteria. So basically most of the gram-negative bacterial species are harmful for humans, and they cause different types of diseases in humans. So there are different types of bacterial infections caused by gram-negative bacteria, which includes like Neisseria gonorrhea, which is also a species of gram-negative bacteria, and it causes gonorrhea, and also known as salpingitis, which is a bacterial infection of urethra and vagina in humans. So in the case of female, it is also called infertility in some cases. The another species is E. coli or Escherichia coli, which causes watery and bloody diarrhea in humans. So it causes sometimes some intestinal diseases or sometimes as extra-intestinal diseases such as urinary tract infections and some other pneumonia and other infections in humans. The another species is Salmonella species, like Salmonella typhi and many other species of the genus, which cause enteric and typhoid fever in human, which includes headache, fever, and many other symptoms in the humans. A species named as Vibrio cholera, which is very common, and it causes cholera disease, which is associated with profuse watery diarrhea with a heavy loss of fluid. Sometimes it also causes death, or it is deadly disease and very serious disease. So there are some most common and most effective disease caused by gram-negative bacteria. So these diseases can be cured by the help of antibiotics, but due to the composition of cell wall composition, there are several antibiotics which cannot affect the gram-negative bacteria, or we can say that gram-negative bacteria can resist the antibiotics. Now let's understand about the treatment. So as we have discussed that they can be treated by the help of antibiotics. Several antibiotics are cephalosporin, which help and which act by the disruption of the cell wall of the particular bacteria. The another antibiotic is tetracycline, which inhibit the protein synthesis, and fluoroquinolones, which inhibit the nucleic acid, which can be DNA or RNA, aminoglycosides, which inhibit the protein synthesis, and streptogramin, which also inhibit the protein synthesis of bacteria. So there are some common antibiotics used against the bacterial infections for gram-negative bacteria.